Hi, so I'm Carina Strell. I'm an associate professor at Uppsala University and researcher at Bergen University, and I'm happy to present you our work on PD1, PD-L1 interactions as a predictive biomarker for immunotherapy benefit in non-small cell lung cancer. So in this project, we hypothesized that the interaction of PD1 and PD-L1 is likely a better biomarker for immunotherapy benefit than measuring PD-L1 protein alone. And in order to measure the interaction of PD1 and PD-L1 in tissue sections, we used a proximity ligation assay from Navinci, the Navini Brightfield kit. And here you see the result. We could nicely detect using the fast red chromogen the interaction sites directly in the tissue contacts. And we apply a QPath analysis pipeline to digitally quantify cells positive for the interaction of PD1 and PD-L1. So after we have set up the assay and had performed control experiments that worked very nicely, we analyzed our first non-small cell lung cancer cohort, which consists of early stage non-small cell lung cancer patients that were surgically treated. And these patients are these samples are in form of a TMA. And this cohort, we stained with the proximity ligation assay for PD1 and PD-L1 interaction, but also for PD1 and PD-L1 protein alone by IHC. And as you see here on this Venn diagram, not all patients, even though they have PD1 and PD-L1 protein interaction, but they are not necessarily positive for the interaction. About 50% of those patients that had both proteins were also positive for the interaction, indicating that the PLA assay really identifies a specific subset of patients. After this, we applied the PLA assay on a pan-cancer cohort, reflecting several different solid tumor types. And you can appreciate from this figure here, we see varying levels of PD1, PD-L1 interactions across these tumor types. And also, on these, the more interactions there are, the higher was the literature-reported objective response rate also for these tumor types. And we thought this was very encouraging, and so we went on and analyzed our immunotherapy-treated non-small cell lung cancer cohort that we have in Uppsala. And we have one part of the cohort is patients treated with immunotherapy, and the other part are patients that are immunotherapy naive. They received chemotherapy. And interestingly, as you can see here, really those patients that had high interaction levels of PD1, PD-L1, they showed a significant longer survival after therapy start than those patients that had very low levels of these interactions. And here in the immunotherapy naive cohort, we didn't see a strong separation, and if at all, patients with high interactions of PD1 and PD-1 showed a worse overall survival. So this really showed that this effect of PD1, PD-L1 interaction is a treatment-specific predictive effect that was also confirmed in formal interaction tests, which remained significant. And also importantly here, in a multivariable model, where we integrated then also histology, stage, age, sex, and smoking history, the PD1, PD-L1 interaction was actually the only variable significantly associated with survival. Finally, of course, we had to test how this observation stands against the clinical standard for the selection of patients to immunotherapy, and that's the PD-L1 tumor proportion score. So PD-L1 expression in tumor cells assessed by IHC. And as one sees, neither a 1% cutoff for this PD-L1 protein nor a 50% cutoff was stratified the patients in a predictive way as we have seen it with the PLA assay. And finally, of course, important to also check how the PD1, PD-L1 interaction status associates with clinical outcome. And here we see the patients of the high group, they had a significant enrichment of complete responses or partial responses as compared to patients with PD1, PD-L1 interactions in a low frequency. And again, also here, the clinical standard of PD-L1 protein failed to show this association or this enrichment. But when one looks at this graph here, it's really nice, but one sees that still there are some patients that have high interaction levels of PD1 and PD-L1, but they still do not respond or benefit from immunotherapy. They have progressive or stable disease. So we were interested in analyzing this group a little bit further, and therefore it was great that we actually had RNA-seq data available for the majority of patients of our immunotherapy cohort. So we performed differentially gene expression analysis, and to cut a long story short, we could identify a set of 13 genes that were specifically upregulated in patients with high PD1, PD-L1 interactions, but no benefit from immunotherapy. So we assume that these proteins that we identified, or these targets that we identified here on the RNA level, are probably likely involved in resistance mechanisms. And it would be, of course, nice now in the future to also analyze these markers in a tissue context. And some of these are alternative immune checkpoint inhibitors, so we would really like to establish a multiplex PLA assay where we can directly integrate these potential resistance mechanisms. Yeah, so in short, what the study could show us is that PD1, PD-L1 interactions, they really serve as a predictive biomarker for benefit from immunotherapy in non-small cell lung cancer patients. And the interaction also outperformed the clinical standard of PD-L1 tumor proportion score via immunohistochemistry.